Chapter 9. Heredity and Evolution. We have seen that reproductive processes give rise to new individuals that are similar, but subtly different. We have discussed how some amount of variation is produced even during asexual reproduction. The number of successful variations are maximized by the process of sexual reproduction. If we observe a field of sugarcane, we find very little variations among the individual plants. But in a number of animals, including human beings, which reproduce sexually, quite distinct variations are visible among different individuals. In this chapter, we shall be studying the mechanism by which variations are created and inherited. The long-term consequences of the accumulation of variations are also an interesting point to be considered. We shall be studying this under evolution. Accumulation of variation during reproduction. Inheritance from the previous generation provides both a common basic body design, and subtle changes in it, for the next generation. Now think about what would happen when this new generation, in its turn, reproduces. The second generation will have differences that they inherit from the first generation, as well as newly created differences. If a single individual divides to reproduce, then the resultant two bacteria divide again. The four individual bacteria generated would be very similar. There would be only very minor differences between them, generated due to small inaccuracies in DNA copying. However, if sexual reproduction is involved, even greater diversity will be generated. Do all these variations in a species have equal chances of surviving in the environment in which they find themselves? Obviously not. Depending on the nature of variations, different individuals would have different kinds of advantages. Heredity. The passing of traits from parents to offsprings is the basis of heredity. Traits are nothing but noticeable characteristics which are transferred from parents to children. There might be certain characteristics which are combination of both the parents. We all inherit traits from our parents and pass them to our children. What exactly do we mean by similarities and differences? We know that a child bears all the basic features of a human being. However, it does not look exactly like its parents. Inherited traits. Have you ever noticed earlobes of different individuals? The lowest part of the ear, called the earlobe, is closely attached to the side of the head in some of us, and not in others. Free and attached earlobes are two variants found in human populations. The rules for inheritance of such traits in human beings are related to the fact that both the father and the mother contribute practically equal amounts of genetic material to the child. Mendel carried out the experiment. He used a number of contrasting visible characters of garden peas, round or wrinkled seeds, tall or short plants, white or violet flowers, and so on. He took pea plants with different characteristics, a tall plant and a short plant, produced progeny from them, and calculated the percentages of tall or short progeny. In the first place, there were no halfway characteristics in this first generation, or F1 progeny, no medium height plants. All plants were tall. Were the tall plants in the F1 generation exactly the same as the tall plants of the parent generation? The progeny of the parental plants are, of course, all tall. However, the second generation, or F2, progeny of the F1 tall plants are not all tall. Instead, 1/4 of them are short. This indicates that both the tallness and shortness traits were inherited in the F1 plants, but only the tallness trait was expressed. Thus, two copies of the trait are inherited in each sexually reproducing organism. These two may be identical, or may be different, depending on the parentage. In this explanation, both capital T's and capital and small t are tall plants, while only small t's is a short plant. In other words, a single copy of capital T is enough to make the plant tall, while both copies have to be small t for the plant to be short. Traits like capital T are called dominant traits, while those that behave like small t are called recessive traits. What happens when pea plants showing two different characteristics, rather than just one, are bored with each other? What do the progeny of a tall plant with round seeds and a short plant with wrinkled seeds look like? They are all tall and have round seeds. Tallness and round seeds are thus dominant traits. But what happens when these F1 progeny are used to generate F2 progeny by self-pollination? A Mendelian experiment will find that some F2 progeny are tall plants with round seeds, and some were short plants with wrinkled seeds. Thus, the tall or short trait and the round seed or wrinkled seed trait are independently inherited. How do these traits get expressed? How does the mechanism of heredity work? Cellular DNA as the information source for making proteins in the cell. A section of DNA that provides information for one protein is called the gene for that protein. How do proteins control the characteristics that we are discussing here? Let us take the example of tallness as a characteristic. We know that plants have hormones that can trigger growth. Plant height can thus depend on the amount of a particular plant hormone. Consider now an enzyme that is important for this process. If this enzyme works efficiently, a lot of hormone will be made, and the plant will be tall. If the gene for that enzyme has an alteration that makes the enzyme less efficient, the amount of hormone will be less, and the plant will be short. Thus, genes control characteristics, or traits. If the interpretations of Mendelian experiments we have been discussing are correct, then both parents must be contributing equally to the DNA of the progeny during sexual reproduction. If both parents can help determine the trait in the progeny, both parents must be contributing a copy of the same gene. This means that each plant must have two sets of all genes, one inherited from each parent. For this mechanism to work, each germ cell must have only one gene set. How do germ cells make a single set of genes from the normal two copies that all other cells in the body have? This is explained by the fact that each gene set is present, not as a single long thread of DNA, but as separate independent pieces, each called a chromosome. Thus, each cell will have two copies of each chromosome, one each from the male and female parents. Every germ cell will take one chromosome from each pair, and these may be of either maternal or paternal origin. When two germ cells combine, they will restore the normal number of chromosomes in the progeny, ensuring the stability of the DNA of the species. Such a mechanism of inheritance explains the results of the Mendel experiments, and is used by all sexually reproducing organisms. But asexually reproducing organisms also follow similar rules of inheritance. How is the sex of a newborn individual determined? Some species rely entirely on environmental cues. Thus, in some animals, the temperature at which fertilized eggs are kept determines whether the animals developing in the eggs will be male or female. In other animals, such as snails, individuals can change sex, indicating that sex is not genetically determined. However, in human beings, the sex of the individual is largely genetically determined. In other words, the genes inherited from our parents decide whether we will be boys or girls. But so far, we have assumed that similar gene sets are inherited from both parents. If that is the case, how can genetic inheritance determine sex? The explanation lies in the fact that all human chromosomes are not paired. Most human chromosomes have a maternal and a paternal copy, and we have 22 such pairs. But one pair, called the sex chromosomes, is odd in not always being a perfect pair. Women have a perfect pair of sex chromosomes, both called X. But men have a mismatched pair in which one is a normal-sized X while the other is a short one called Y, so women are XX, while men are XY. Now, can we work out what the inheritance pattern of X and Y will be? Half of the children will be boys and half will be girls. All children will inherit an X chromosome from their mother regardless of whether they are boys or girls. Thus, the sex of the children will be determined by what they inherit from their father. A child who inherits an X chromosome from her father will be a girl, and one who inherits a Y chromosome from him will be a boy. Evolution. We have noted that there is an inbuilt tendency to variation during reproduction, both because of errors in DNA copying, and as a result of sexual reproduction. Let us now look at some consequences of this tendency. Consider a group of 12 red beetles. They live, let us assume, in some bushes with green leaves. Their population will grow by sexual reproduction, and therefore, can generate variations. Let us imagine also that crows eat these beetles. The more beetles the crows eat, the fewer beetles are available to reproduce. Now, let us think about some different situations that can develop in this beetle population. If a color variation arises during reproduction, so that there is one beetle that is green in color instead of red, this beetle, moreover, can pass the color on to its progeny, so that all its progeny beetles are green. Crows cannot see green-colored beetles on the green leaves of the bushes, and therefore cannot eat them. What happens then? The progeny of green beetles is not eaten, while the progeny of red beetles continues to be eaten. As a result, there are more and more green beetles than red ones in the beetle population. In a second situation, again, a color variation arises during reproduction, but now it results in a beetle that is blue in color instead of red. This beetle can also pass the color on to its progeny, so that all its progeny beetles are blue. Crows can see blue-colored beetles in the green leaves of the bushes as well as they can see red ones, and therefore can eat them. What happens initially? In the population, as it expands, there are a few blue beetles, but most are red. But at this point, an elephant comes by, and stamps on the bushes where the beetles live. This kills most of the beetles. By chance, the few beetles that have survived are mostly blue. The beetle population slowly expands again, but now, the beetles in the population are mostly blue. It is obvious that in both situations, what started out as a rare variation came to be a common characteristic in the population. In other words, the frequency of an inherited trait changed over generations. Since genes control traits, we can say that the frequency of certain genes in a population changed over generations. This is the essence of the idea of evolution, but there are interesting differences too, in the two situations. In the first case, the variation became common because it gave a survival advantage. We can see that the natural selection is exerted by the crows. The more crows there are, the more red beetles would be eaten, and the more the proportion of green beetles in the population would be. Thus, natural selection is directing evolution in the beetle population. It results in adaptations in the beetle population to fit their environment better. In the second situation, the color change gave no survival advantage. Instead, it was simply a matter of accidental survival of beetles of one color that changed the common characteristic of the resultant population. The elephant would not have caused such major havoc in the beetle population if the beetle population had been very large. So, accidents in small populations can change the frequency of some genes in a population, even if they give no survival advantage. This is the notion of genetic drift, which provides diversity without any adaptations. Now consider a third situation. In this, as the beetle population begins to expand, the bushes start suffering from a plant disease. The amount of leaf material for the beetles is reduced. The beetles are poorly nourished as a result. The average weight of adult beetles decreases from what it used to be when leaves were plentiful, but there is no genetic change occurring. After a few years and a few beetle generations of such scarcity, the plant disease is eliminated. There is a lot of leaf food. At this time, what would we expect the weight of the beetles to be? The beetles would again nourished, and as a result the average weight of adult beetles would increase. Acquired and inherited traits. We discussed the idea that the germ cells of sexually reproducing populations are made in specialized reproductive tissue. We have seen the beetle population in the third situation. Change in non-reproductive tissues cannot be passed on to the DNA of the germ cells. Therefore, the experiences of an individual during its lifetime cannot be passed on to its progeny, and cannot direct evolution. Consider another example of how an individual cannot pass on to its progeny the experiences of its lifetime. If we breed a group of mice, all their progeny will have tails, as expected. Now, if the tails of these mice are removed by surgery in each generation, do these tailless mice have tailless progeny? The answer is no, and it makes sense because removal of the tail cannot change the genes of the germ cells of the mice. Charles Robert Darwin. Charles Darwin set out on a voyage when he was 22 years old. The 5-year voyage took him to South America and the islands off its coast. The studies that he conducted during this voyage were to change forever the way we look at the variety of life on Earth. He formulated his hypothesis that evolution took place due to natural selection. He did not know the mechanism whereby variations arose in the species. He would have been enlightened by Mendel's experiments, but these two gentlemen did not know of each other or their work. Speciation. Let's think of group of beetles. If it splits into two populations that cannot reproduce with each other. When this happens, they can be called two independent species. So, can we extend the reasoning we have used above to explain such speciation? Consider what would happen if the bushes the beetles feed on are spread widely over a mountain range. The beetle population becomes very large as a result. But individual beetles feed mostly on a few nearby bushes throughout their lifetime. They do not travel far. So, in this huge population of beetles, there will be subpopulations in neighborhoods. Since male and female beetles have to meet for reproduction to happen, most reproduction will be within these subpopulations. Of course, an occasional adventurous beetle might go from one site to another. Or a beetle is picked up by a crow from one site and dropped in the other site without being eaten. In either case, the migrant beetle will reproduce with the local population. This will result in the genes of the migrant beetle entering a new population. This kind of gene flow is bound to happen between populations that are partly, but not completely separated. If, however, between two such subpopulations a large river comes into existence, the two populations will be further isolated. The levels of gene flow between them will decrease even further. Over generations, genetic drift will accumulate different changes in each subpopulation. Also, natural selection may also operate differently in these different geographic locations. Thus, for example, in the territory of one subpopulation, crows are eliminated by eagles. But this does not happen for the other subpopulation, where crow numbers are very high. As a result, the green variation will not be selected at the first site, while it will be strongly selected at the second. Together, the processes of genetic drift and natural selection will result in these two isolated subpopulations of beetles becoming more and more different from each other. Eventually, members of these two groups will be incapable of reproducing with each other even if they happen to meet. There can be a number of ways by which this can happen. If the DNA changes are severe enough, such as a change in the number of chromosomes, eventually the germ cells of the two groups cannot fuse with each other. Or a new variation emerges in which green females will not mate with red males, but only with green males. This allows very strong natural selection for greenness. Now, if such a green female beetle meets a red male from the other group, her behavior will ensure that there is no reproduction between them. Effectively, new species of beetles are being generated.
Evolution and its theory. Evolution is considered as the process of gradual change occurring in living organisms over a long duration. In other words, evolution is considered as the process of formation of new organisms from primitive ones by slow changes. Theory of evolution suggests that living organisms originated in the ocean. Initially, unicellular organisms formed, which gradually evolved into multicellular and complex organisms. It took around 300 crore years for these changes to occur. These changes have been multidimensional in living organisms, which led to evolution of various kinds of living organisms.
Fossils. How do we know that extinct species ever existed? We know this from finding fossils. What are fossils? Usually, when organisms die, their bodies will decompose and be lost. But every once in a while, the body or at least some parts may be in an environment that does not let it decompose completely. If a dead insect gets caught in hot mud, for example, it will not decompose quickly, and the mud will eventually harden and retain the impression of the body parts of the insect. All such preserved traces of living organisms are called fossils. If we dig into the earth and start finding fossils, it is reasonable to suppose that the fossils we find closer to the surface are more recent than the fossils we find in deeper layers. The second way of dating fossils is by detecting the ratios of different isotopes of the same element in the fossil material. It would be interesting to find out exactly how this method works.
Fossils are often used as evidence of evolution. Fossils are considered as remains of dead organisms that lived long ago. Several organisms get buried due to disasters like flood, earthquake, volcano, etc. Remnants and impressions of such organisms remain preserved underground. Once the organism dies, carbon consumption stops and decaying process of C14 starts. The time passed since the death of a plant or animal can be calculated by measuring the radioactivity of C14 and ratio of C14 to C12 present in their body. This is known as carbon dating method. This method is used in paleontology and anthropology for determining the age of human fossils.
Evolution by stages. A question that arises here is, if complicated organs, such as the eye, are selected for the advantage they provide, how can they be generated by a single DNA change? Surely such complex organs will be created bit by bit over generations? But how can each intermediate change be selected for? There are a number of possible explanations. Even an intermediate stage, such as a rudimentary eye, can be useful to some extent. This might be enough to give a fitness advantage. In fact, the eye like the wing seems to be a very popular adaptation. Insects have them, so does an octopus, and so do vertebrates. And the structure of the eye in each of these organisms is different enough for them to have separate evolutionary origins. To say that very dissimilar looking structures evolved from a common ancestral design. It is true that analysis of the organ structure in fossils allows us to make estimates of how far back evolutionary relationships go. But those are guesses about what happened in history. Are there any current examples of such a process? The wild cabbage plant is a good example. Humans have over more than 2000 years cultivated wild cabbage as a food plant, and generated different vegetables from it by selection. This is, of course, artificial selection rather than natural selection. So some farmers have wanted to select for very short distances between leaves, and have bred the cabbage we eat. Some have wanted to select for arrested flower development, and have bred broccoli, or for sterile flowers, and have made the cauliflower. Some have selected for swollen parts, and come up with kohlrabi. Some have simply looked for slightly larger leaves, and come up with a leafy vegetable called kale. Another way of tracing evolutionary relationships depends on the original idea that we started with. That idea was that changes in DNA during reproduction are the basic events in evolution. If that is the case, then comparing the DNA of different species should give us a direct estimate of how much the DNA has changed during the formation of these species.